Bismillahirrahmanirrahim. Welcome to Forward Public School E-Learning Class 9th, Subject Biology, Chapter number 5, Cell Cycle. I am your teacher Seema Samin. Students, in the previous class we have started cell cycle and we have done briefly about stages or phases of cell cycle, that is interphase and division phase. We have done interphase in detail. So today we are going to start division phase. For that you must have pencils in your hand to underline difficult words and write down their meanings. This is the second topic of the chapter, that is cell cycle. So here we will be discuss division phase in detail. Division phase, that is done in two ways, or there are two types of cell division, that is number one mitosis and number second is meiosis. We will discuss these two processes in details. So first come towards mitosis. Students, for division phase come to page number 102 of your book. डिविजन फेज देखें आप. ड्यूरिंग द डिविजन फेज द सेल अंडरगोज डिविजन आइदर बाय माइटोसिस और मेयोसिस. जब किसी सेल ने डिविजन करनी है तो डिविजन विल बी डन बाय टू वेज और टू टाइप्स. सेल डिविजन करेगा या तो माइटोसिस से या फिर मेयोसिस से. सो लेट्स डिस्कस दीज टू टाइप्स वन बाय वन. फर्स्ट कम टूवर्ड्स माइटोसिस. अ जर्मन बायोलॉजिस्ट नेम्ड वाल्टर फ्लेमिंग observed a dividing cell. He noticed that during division, nucleus passes through a series of changes. He called these changes as mitosis. In this type of cell division, the cell divides into two daughter cells. In each daughter cell, the chromosome number is maintained at the same level as that in the parent cell. वाल्टर फ्लेमिंग ने फर्स्ट टाइम जो है डिवाइडिंग सेल को ऑब्जर्व किया कि जब कभी सेल डिवाइड हो रहा हो तो उस दौरान क्या चेंजेस आती है सेल में. सो ड्यूरिंग डिवीजन न्यूक्लियस जब देखा गया सो उसमें डिफरेंट चेंजेस आए, डिफरेंट स्टेप्स में वो चेंजेस आए विच आर कॉल्ड सीरीज ऑफ चेंजेस. सो इन सच वे दैट प्रोसेस इज कॉल्ड माइटोसिस. इन दिस टाइप ऑफ सेल डिवीजन जो माइटोसिस टाइप है, इसमें एक सेल डिवाइड होकर बनाता है टू डॉटर सेल्स. एक सेल से दो सेल्स बनते हैं और ईच डॉटर सेल, हर एक डॉटर सेल में द क्रोमोसोम नंबर इज मेंटेन एट द सेम लेवल एज दैट इन द पेरेंट सेल. और दो सेल जो एक सेल के डिवीजन के रिजल्ट में बनते हैं, उन दो में क्रोमोसोम नंबर सेम ही होता है जैसे कि पेरेंटल सेल में हो. सो सिंपली हम डिफाइन कर सकते हैं माइटोसिस को. इट इज़ अ टाइप ऑफ सेल डिवीजन in which daughter cell receive the same number of chromosomes as present in the parental cell. दोबारा सुने. A type of cell division in which daughter cell receive the same number of chromosomes as present in the parental cell. Scientific information को आप देखें. Mitosis take place in eukaryotic cell, whereas in prokaryotic cell binary fission take place, which is called amitosis. Binary fission आप देखें. Binary means two. And fission means splitting. So splitting of one cell into two. There is a difference between mitosis and amitosis or binary fission. Mitosis जो है it is concerned with nucleus. So it going to be take place in eukaryotic cell. And जो prokaryotic cell है उधर कोई proper nucleus exist नहीं करता, just DNA exist करता है. तो DNA going to be divided by a method which is called amitosis or simply binary fission. Point to ponder आप देखें. The prokaryotes do not undergo mitosis. Why? So the simple answer is there is no proper nucleus existence. That is why prokaryotic cell divided by binary fission as compared to mitosis. Students, come to page number 103 of your book. Come toward stages of mitosis. कि माइटोसिस जो है हाउ इट इज़ गोइंग टू बी डन, व्हाट आर द डिफरेंट स्टेप्स और स्टेजेस विच आर गोइंग टू बी कंसर्न विद माइटोसिस. फॉर कन्वीनियंस द प्रोसेस ऑफ माइटोसिस कैन बी डिवाइडेड इनटू टू मेन स्टेजेस, कैरियोकाइनेसिस एंड साइटोकाइनेसिस. कोई भी सेल जब माइटोसिस से पास होता है तो दो मेन स्टेजेस या फेजेस से गुजरता है, जिसमें से फर्स्ट कैरियोकाइनेसिस है और सेकंड साइटोकाइनेसिस है. Then finally cell divided into two cells or two daughter cells as we done in the definition. So आप देखिए karyokinesis, karyo it is the division of the nucleus, that is karyo means nucleus and kinesis mean division. So जब cell ने divide होना है through mitosis, division always start from nucleus. So first step will be the division of nucleus, which is called karyokinesis, and second step or stage is cytokinesis, which is the division of cytoplasm. So first we will be discuss karyokinesis, then we will be moved toward cytokinesis. So finally one cell will be divided into two daughter cells. Come towards scientific information. During S phase of interphase, the parent cell makes a copy of each chromosome. In mitosis, the copies of chromosomes are distributed equally to both daughter cells. ठीक है, अगर आपने जैसे लास्ट टॉपिक में किया कि इंटरफेज में जो सेल्स फर्स्ट जो पास होगा G1 फेज से या इंटरफेज के फर्स्ट स्टेप से, तो उसमें क्रोमोसोम इस फॉर्म में थे. बट वेन एवर द सेल पास थ्रू S फेज, द क्रोमोसोम्स गेट डुप्लीकेटेड. क्रोमोसोम्स क्या हो गए? डुप्लीकेटेड हो गए, हर क्रोमोसोम डबल हो गए. एंड फाइनली आफ्टर द इंटरफेज, the cell when divide through division phase, the each and every cell, like the two cells, they will be receive the same number of chromosome. First will be moved to one cell, and the two chromosome will be moved to the opposite cell. So as a result, from one cell, two cell will be produced. Each will be having the same number of chromosome as present in the parental cell. So now come towards karyokinesis. It is further divided into four phases or stages or steps, that is prophase, metaphase, anaphase and telophase. Karyokinesis को हम 4 steps में divide करते हैं: prophase, metaphase, anaphase and telophase. So let's discuss these steps or stages one by one. First come towards prophase. Prophase पे आप आ जाए. So let's start from prophase. Prophase in the cell completed in 2 steps or stages. Cells में prophase 2 steps या stages में complete होता है, जिसमें से the first is called early prophase and the last is called late prophase. So now come towards the changes which are taking place in a cell during prophase. Nucleus में क्या क्या changes आती है during prophase में? Early prophase आप देखें, ये आप देखें, this cell going to be divided. Karyokinesis इस cell में होने जा रहा है. So first come towards chromosomes. Nucleus में chromosomes देखें, they become thicken, they become condense. Now they are called chromosomes. जब cell dividing नहीं था तो ये इस form में exist कर रहा था. In the nucleus, the chromosomes were in the form of fine thread-like structure. They were called chromatin network or chromatin material. But now, when the cell is dividing, they become condensed, they become thick, they become shortened, and now they are called chromosomes. So, during prophase, the chromatin thread condense, coil, and shorten to become chromosomes. Nucleus के अंदर chromatin material कैसे हो जाएगा? Condense हो जाएगा, coil हो जाएगा और shorten हो जाएगा, and now they are called chromosomes. Each chromosome, इसमें से अगर आप कोई एक उठा ले, किसी एक को देख ले, so each chromosome now have a structure where there is the central button-like structure present and having arm, one arm and second arm. The central button is called centromere and these arm-like structure they are called chromatids. ठीक है? So each chromosome consists of 2 sister chromatids attached at the centromere. Centromere and 2 sister chromatid, 1 and 2, first and second. Now this is the complete structure of chromosome. The chromosomes are now visible under a microscope. इस स्टेज पे क्रोमोसोम विजिबल हो जाते हैं, माइक्रोस्कोप में इजीली दिखाई देते हैं. ठीक है, सो दिस इज द अर्ली प्रोफेज where the chromosomes become condensed, coiled and shortened, and they are called chromosomes. They attain a proper shape where there is a central button-like structure present which is called centromere and having arms, 2 arms which are called chromatids. ठीक है, लेट प्रोफेज पे आप आ जाए. इन एन एनिमल सेल The two pairs of centrioles reach opposite poles of the cell. अगर ये एनिमल सेल है तो ये आप देखिए ग्रीन स्ट्रक्चर आपको नज़र आ रहे हैं यहाँ से. वन इज एग्जिस्टेड हेयर, द अदर स्टार्ट ट्रैवलिंग टू मूव टू अपोजिट साइड. ये स्ट्रक्चर जो है, दीज ऑर्गेनल्स दे आर कॉल्ड सेंट्रियोल्स. इन द एनिमल सेल्स दे आर प्रेजेंट. जब सेल ने डिवाइड होना है सो अर्ली प्रोफेज पे ही द वन रिमेन ऑन वन पोल एंड द अदर स्टार्ट मूवमेंट टू और ट्रैवलिंग टू द अपोजिट साइड. ठीक है. सो इन एन एनिमल सेल द टू pairs of centrioles reach opposite pole of the cell. They make a network of spindle fiber between the poles. आप देखो, there are thread-like structure arise from each centriole and that is existed in the cell in the form of a network. The complete set of spindle fiber is called mitotic apparatus. अब इस complete set को centriole along with these fiber-like structure or spindle fiber, this is called mitotic apparatus. अब in the plant cell there are no centrioles, so whenever there is no centriole, their mitotic spindle is formed by aggregation, by the deposition, by the joining, by the attachment of spindle fibers present in the cytoplasm. कि जब प्लांट सेल में सेंट्रियोल एग्जिस्ट ही नहीं कर रहे हैं तो वहाँ माइटोटिक स्पिंडल कैसे बनेंगे. वहाँ जो वेसिकल्स होंगे इन द साइटोप्लाज्म, दे विल बी कंबाइन एंड दे विल बी एग्जिस्टेड ऑन द अपोजिट पोल्स एंड दे विल बी एक्ट लाइक अ माइटोटिक apparatus and fibers will be arise from them. As a part of changes of prophase, the nucleolus disappear. अब आप देखो, here in the nucleus there are some rounded structure present which are plural called alveoli and singular called alveolus. So whenever the nuclear membrane is going to be disappear, the nucleolus is going to be dispersed in the cytoplasm. nucleus का जो envelope cover ख़त्म हो गया है, so the nucleolus dispersed in the cytoplasm and the nuclear envelope breaks down. और nuclear envelope भी जो यहाँ exist कर रही थी that gets broken down and existed in the form of vesicles in the cytoplasm now. Students, now come toward second phase of karyokinesis, that is metaphase. Metaphase पे आप आ जाए. In metaphase, some spindle fibers attached with the chromosomes. In the metaphase, some spindle fibers attached with the chromosomes. क्रोमोसोम्स के साथ माइटोटिक apparatus से जो स्पिंडल फाइबर अराइज होंगे, दे विल बी गेट्स अटैच विद द क्रोमोसोम्स. टू स्पिंडल फाइबर्स फ्रॉम बोथ साइड्स अटैच विद वन क्रोमोसोम. आप एक क्रोमोसोम को देखें, एक क्रोमोसोम के साथ दोनों साइड से अगर ये क्रोमोसोम है, सो फ्रॉम बोथ साइड, फ्रॉम द अबव, फ्रॉम द बिलो, फ्रॉम द अपर वन पोल, फ्रॉम द सेकेंड पोल, वन स्पिंडल फाइबर विल बी अटैच फ्रॉम ईच साइड. The chromosomes attached with the spindle fibers arrange themselves at the equator of the cell, that is in the center of the cell. In this way, a plate is formed called metaphase plate. So, in such way, आप देखें, the chromosomes get attached in the center of the cell. Now they are exist in the form of plate-like structure, which is called metaphase plate. Each chromosome is attached from both sides, from both pole by two fibers. आप देखें, हर एक क्रोमोसोम की अटैचमेंट एक साइड से भी सेंट्रियोल से हुई है थ्रू स्पिंडल फाइबर और दूसरी तरफ से भी सेंट्रियोल से हुई है थ्रू स्पिंडल फाइबर. At this stage, it is called metaphase plate, where chromosomes arrange themselves in the center. आप देखो, यहाँ पे क्रोमोसोम्स जस्ट फ्री हुए थे फ्रॉम द न्यूक्लियस. नाउ दे गेट्स अटैच इन द सेंटर. मेटाफेज पे क्रोमोसोम सेंटर में अटैच हो जाएंगे या फिर अरेंज हो जाएंगे, मेकिंग अ प्लेट लाइक स्ट्रक्चर विच इज कॉल्ड मेटाफेज प्लेट. Students, now come toward page number 104 of your book and come toward third step or stage of karyokinesis, which is called anaphase. As we have done in the metaphase, where chromosomes arrange themselves in the center, making metaphase plate. Now come towards anaphase. Anaphase also completed in two steps or stages, that is early anaphase and late anaphase. So let's come towards early anaphase. That what are the changes which are going to be take place during anaphase. So the centromere of each chromosome split. During anaphase, the chromosomes which were existed in this way, now the centromere will be get split. The centromere will be get split. So in such way, each chromosome will be get divided, and the half chromosome will be existed in this way, half will be existed in this way. So the centromere of each chromosome splits. The spindle fibers pull the chromatids apart to opposite poles. Two chromatids which will be produced, they will be pulled to opposite poles. एक क्रोमैटिड एक पोल पे चला जाएगा और दूसरा क्रोमैटिड दूसरे पोल पे चला जाएगा. They will be pulled by spindle fibers toward the centriole. Once the chromatid are separated, जब chromatids अलग हो गए, separate हो गए, they are called daughter chromosomes. Now they are not called chromatids further, they are called daughter chromosomes. In early anaphase, the centromere splits. In early anaphase, in the first step, the centromere splits. Half the chromosomes move to one pole and half to the other pole. So each chromosome, हर एक क्रोमोसोम का एक हाफ एक पोल पे जाएगा, दूसरा हाफ दूसरे पोल पे, एक हाफ एक पोल पे, दूसरा हाफ दूसरे पोल पे. In such way, all the chromosomes will be get divided and each half will be moved to each pole. Late anaphase पे देखें. In the late anaphase, the chromosomes have almost reached their respective poles. The chromosomes reach their respective poles. They reach toward the point where the centromere, the centrioles are existed. The cell membrane begins to move inward at the center. Looks here, the cell membrane going to be get pinched from the center. That is, it is going to be, now division is going to be started. That is division of cytoplasm. That is the cytokinase is going to be started here. Students, now come towards fourth step or stage of karyokinesis, that is telophase. So in telophase, the spindle fiber breaks down. टेलोफेज के दौरान स्पिंडल फाइबर ख़त्म हो गए. अब आपको ये देखें इस तरह की तरह नज़र नहीं आ रहे. ठीक है. अ न्यूक्लियर एनवेलप फॉर्म्स अराउंड द क्रोमोसोम्स. क्रोमोसोम के इर्द गिर्द क्या बन गया है? न्यूक्लियर एनवेलप, जो कि प्रोफेज के दौरान ख़त्म हुआ था. नाउ इट्स अगेन प्रोड्यूस्ड. अ न्यूक्लियर एनवेलप फॉर्म्स अराउंड द क्रोमोसोम्स एट ईच पोल ऑफ द cell. आप देखो cell is still one cell, but the chromosome move to opposite poles, and on these opposite poles around the chromosome nuclear envelope is produced. Thus the two daughter nuclei are formed in each cell, or in the one cell two daughter nuclei are formed now, each with the same number of chromosomes, each with the same number of chromosomes as were present in the parent nucleus. A nucleolus formed inside the nuclei. Each nucleolus will be formed and chromosome uncoiled to become thin chromatin network, and again chromosome will be get coiled. They become very thin thread-like structure now, and at this stage still the cell is the one. It is not divided. It is not divided by cytoplasm. So further, or the second step, the second step or the second main stage will be cytokinesis. So let's move toward cytokinesis. Cytokinesis, which is simply the division of cytoplasm whenever done. So a single cell divided into two daughter cells, which is clearly visible in this diagram. आप देखें, after the division of nucleus, the cytoplasm divides. The division of cytoplasm is called cytokinesis. In animal cell and in plant cell, cytokinesis occur in a bit different way. In animal cell, cleavage or furrow appear in the cytoplasm between the two nuclei. In the animal cell, there is a cleavage appear in the center, which is called cleavage or furrow. And as a result, whenever it get deepened from both sides, so one cell divided into two daughter cells. The furrow deepen, and two identical or same daughter cells are finally produced. In the plant cell, this division is a bit different. In plant cell, a cell plate is formed between the two daughter nuclei. In the plant cell, there is in the center of the cell a plate-like structure formed which is called cell plate, and this cell plate leading to the division of one cell into two daughter cells. It divides the cell into two. As a result of formation of this plate, the cell divided into two daughter cells. The cell plate is formed by the fusion of small fluid-filled vesicles produced by Golgi apparatus. This cell plate-like structure, it is the aggregation or it is the combination of different vesicle-like structure which are produced as a result at the margins of the Golgi apparatus, and finally they help in the division of cell into two daughter cell. So as a result, like in this way, there is a difference existed between the division of cytoplasm in the plant cell as well as in the animal cell. Animal cell divided by furrow formation or cleavage formation, and plant cell divided by vesicle formation. Students, with the end of cytokinesis, the process of mitosis is completed. I hope you understand it well. Now come towards significance of mitosis, that what are the importance of mitosis, how this process is helpful in our daily life, or what type of significance we have from the process of mitosis. So here we have some significance of mitosis, some roles of mitosis in the life of an organism. The first one is growth. Growth simply the increase in size or number. For a multicellular organism, that is organism having more number of cells, so multicellular organisms to grow, new cells are produced by mitosis. That is, the organism size is totally dependent on mitosis, because whenever mitosis is done, from 1 cell 2 to 4, 4, 8, such type of cells going to produce by the process of mitosis. So mitosis helpful है हमारी growth dependent है किस पे, mitosis, because mitosis produce same type of cells. So हमारी body में हमारा जो size increase हो जाता है, height, each and everything, it is dependent on mitosis. Second आप देखिए, repair of worn out parts, that is unwanted parts. Each time you brush your teeth and rinse your mouth, hundreds of dead and worn out cheek cells are being shed into your saliva, that is water in the mouth. These dead cells are constantly being replaced by the process of mitosis. हर वक्त जो हमारी माउथ में या हमारी टीथ या इन एरिया से जो सेल्स, जो डेड सेल है वो शेड ऑफ हो जाते हैं, वो रिमूव हो जाते हैं, तो इनको जो कंटिन्यूसली या फिर अगेन एंड अगेन रिप्लेस करते हैं न्यू सेल्स, वो कौन से प्रोसेस से होता है, बाय माइटोसिस. तो इस तरह अनवांटेड सेल्स का जाना और न्यू सेल्स का आना, इट इज़ ऑल्सो डिपेंडेंट ऑन माइटोसिस, और सिंपली इट इज़ ऑल्सो डन बाय माइटोसिस. थर्ड आप देखिए एसेक्सुअल रिप्रोडक्शन. एसेक्सुअल रिप्रोडक्शन दैट इज़ द increase in number by the increase in the organisms by the method which is called mitosis, called asexual reproduction. Many organisms reproduce asexually, that is there is a single parent is going to be single parent is capable to be give rise to organism or new ones or young ones. For example, cutting, grafting, budding, etc. These are the different methods of asexual reproduction. Hydra, a marine animal, reproduce asexually by budding. हाइड्रा एक मेरीन एनिमल है जो कि कैसे रिप्रोड्यूस होता है, बडिंग के प्रोसेस से. बड आर एन आउटग्रोथ, बड्स आर आउटग्रोथ विच अराइज ऑन द बॉडी, एंड विद द पैसेज ऑफ टाइम दे गेट्स डिटैच फ्रॉम द बॉडी एंड स्टार्ट देयर independent life. During this process, that is during budding process, mitosis forms a mass of cells called bud on the surface of a hydra. On the body of hydra, there is a mass of cell developed in the form of outgrowth or bud-like structure, and this bud is finally detached and start its independent life. So this is simply done by mitosis. The mitosis continue in the cells of bud and it grow into a new hydra. Flowering plants, that is flower producing plants, are capable of vegetative reproduction through the process of mitosis in their vegetative parts, that is shoots and roots. रूट्स का, शूट का, स्टेम का, लीव्स का, इन सब का भी बढ़ना, दैट इज़ इंक्रीज इन नंबर और साइज, इट इज़ ऑल्सो डिपेंडेंट ऑन माइटोसिस. दिस शूट, रूट, दे आर वेजिटेटिव पार्ट. रीजनरेशन, it is the process of renewal of organisms or the worn out cells and tissues. Regeneration, like whenever a part of the body is removed, so it's going to be regenerate, it's going to be reproduce. So it is also dependent on mitosis. सो इस प्रोसेस में रिन्यूअल ऑफ ऑर्गेनिज्म या फिर बॉडी का कोई पार्ट जो चला गया है उस पार्ट की रिन्यूअल होता है या वो अगेन प्रोड्यूस होता है. दिस इज कॉल्ड रीजनरेशन. इट इज़ ऑल्सो डन बाय माइटोसिस. All organisms have the ability to regenerate cells and tissues that are broken due to certain processes such as necrosis, that is death of cells. ठीक है, necrosis is death of cells. For example, regeneration of tail in lizards. लिजर्ड्स में जो टेल है, अगर टेल रिमूव हो जाए, कहीं कट जाए, कटिंग हो जाए टेल की, तो लिजर्ड्स हैव द पावर टू बी रीजनरेट और टू अगेन प्रोड्यूस देयर टेल, विच इज़ एन एग्जाम्पल ऑफ रीजनरेशन. Other organisms have the ability to regenerate the whole body from a piece of the body. For example, Hydra. ठीक है, इस तरह यहाँ हमने देख लिया कि एक बर्ड जो है, whenever the bird will be get detached, it will be going to be changed into a complete organism. So these are the multiple significance of mitosis. There are much more other benefits also existed, which are existed in the organism's life. Students, this was the end of our topic. Next topic of the chapter will be discussed in the next class. Thank you.